Good morning. Thank you for joining us today for the first public briefing of the Massachusetts Consortium on Pathogen Readiness. I'm George Daley, the Dean of Harvard Medical School. In late December, clusters of patients with an unusual pneumonia started presenting at hospital emergency rooms in the city of Wuhan, China. Within weeks, the culprit was identified as a novel coronavirus, SARS-CoV-2. By then, the pathogen had spread with stunning infectivity to other parts of the globe. It was clear that the impact of the virus would be widespread and have devastating consequences. In late January, Chairman Hui Yakan of the China Evergrande Group, a generous supporter of the Evergrande Center for Immunologic Diseases at the Harvard Medical School and the Brigham and Women's Hospital, sent a letter to President Larry Bacow requesting scientific and clinical support for confronting the new virus. Feeling a great responsibility to respond to this humanitarian crisis, President Bacow charged Alan Garber and me to convene our community to offer help. In the ensuing weeks, as COVID-19 spread throughout the world, including here in the United States, experts across a spectrum of disciplines began communicating with a team in Guangzhou, China, and we began learning essential facts about the novel COVID disease from our Chinese colleagues who were on the front lines of the epidemic. On March 2nd, Harvard Medical School convened a meeting to bring together key people across the Commonwealth and our colleagues in China to organize a more formal, integrated effort to confront the disease, and MassCPR was born. MassCPR has now become a multi-institutional collaboration encompassing hundreds of scientists, clinicians, and public health experts based in Massachusetts, working collaboratively alongside international colleagues who have joined to tackle this global threat on behalf of humanity. What I've witnessed over these last 3 months has been truly humbling. I've seen our investigators placing community benefit and global health above individual, institutional, and corporate interests. By bringing us together, this tragedy brings out the best in us. We cannot deal with a global threat by going it alone or by being isolationist. We must work together across labs, across institutions, across geographic borders. We are doing this through MassCPR. I cannot think of a better example of regional and transnational collaboration against a common enemy. When we act together, we are all better off individually and collectively. So we are forging ahead with full steam towards two goals: to turn the tide of this current pandemic and to create a durable rapid response system to address future health crises. I'm immensely proud of the work our growing consortium, and I am excited that our speakers today will have a chance to share details about their progress. First, allow me to welcome two people who have been catalytic to our work. MassCPR was enabled by a collaborative agreement between Harvard University, the Guangzhou Institute of Respiratory Health, and the China Evergrande Group. Dr. Zhong Nanshan is leading the collaborative efforts at the Guangzhou Institute of Respiratory Health. He is a hero of the SARS outbreak of 2003 and a bona fide legend in the field of public health, epidemiology, and pulmonology. His expertise and insights have been invaluable to us as well as to the entire global scientific community. Welcome, and I invite some comments from Dr. Zhong.
Good morning, George, David, Alan, James, and the expert of the Massachusetts Consortium Pathogen Readiness. Ladies and gentlemen, good morning. So on behalf of the Guangzhou Institute of Respiratory Health, and here, all of us, I would like to express my many thanks for being able to attend this MassCPR meeting, so public briefing. So I remember on 29th of January So that was at the beginning of the global pandemic of COVID-19 infection. I got a phone call from Mr. Jiayin Xu and telling me he would like to support a joint research program for the control and management of against COVID-19 infection between Harvard Medical School and Guangzhou Institute of Respiratory Health, with 5 years with funding of 800 million RMB. That means the 110 million US dollars in the next 5 years. Based on his arrangement, we had the meeting. I and my team have a meeting with George and your team. So the president of Harvard Medical School on the 4th of February. So since then, we have already had 11 times of webinar meeting discussing and sharing our experience on and the lessons on fighting against COVID-19 and discussing about how we can arrange the program. So, and the so after 5 rounds of the evaluation of the applicants' topics, so from both sides, so we have worked out more than 20 recommended proposals from the Harvard side is 28, from our side is 22, and some key research resources. So actually they are characterized as, firstly, the proposals of both sides are more oriented to clinical practice, including disease protection and epidemiology, and develop fast and simple tests of diagnostic tests, and so drug management and vaccine. But I think part of them, the proposals, may be completed within 2 years. So that's first. And secondly, part of the proposals can be cooperated by both sides, such as simple and fast diagnostic tests and off-label medication for the management of COVID-19, and monoclonal antibody development, a multi-center trial, and something like a deep learning electronic medical records and CT scan for diagnosis and grading and outcome prediction, and so on. So far, our collaboration is going smoothly and fruitfully. So I really appreciate the opportunity of that Guangzhou Institute of Respiratory Health is able to cooperate with the Harvard Medical School, one of the top medical schools in the world. So actually, from my part, so we yesterday, we had worked out 22 proposals and discussed from each other. Majority of the PI actually have been trained in the US, I should say that. So, and in the meantime, I would like to express many thanks to Mr. Xu and James from Evergrande, who have made the cooperative proposals into real practice. So let's work together. I'm sure the scientists and the clinicians from both sides may offer a great contribution in combating the pandemic COVID-19 infection. And if we have got the further achievement, so therefore we are able to get the continuous support from Evergrande. I'm sure that the substantial cooperation between us will be of great success. Thank you very much.
Thank you very much, Dr. Zhong. Our relationship has truly been inspiring. Thank you so much for your leadership. Let me now introduce CEO James Xia of the China Evergrande Group, which is providing funding support to researchers based both in Massachusetts and in China to combat COVID-19. James joins us from China. Welcome, James.
Thank you. Dear Dean Daley, Dr. Zhong, distinguished experts from MassCPR, and friends from the press. Good morning. It is my great pleasure to communicate with you online. First of all, on behalf of China Evergrande Group, I would like to extend my warmest congratulations to the official formation of MassCPR and express my highest regards to scientists and clinicians in both China and the US who are working together to fight coronavirus pandemic. Earlier this year, from late January to February, entrusted by Mr. Hui Gayin, chairman of China Evergrande Group, I led my team to visit Harvard University to meet with President Barouch, Provost Sharpe, Dean Daley, and many others. We will be able to discuss the ongoing epidemic conditions then in China in great detail and explore joint efforts in research cooperation to combat the epidemic. Thanks to everyone who rose to the call of action. With just 2 short weeks, we'll be able to establish the 3 parties cooperation among Harvard University, Guangzhou Institute of Respiratory Health, and China Evergrande. China Evergrande would provide 800 million RMB to over the next 5 years to support leading scientific and medical experts from the 2 countries to develop diagnostics tests, vaccines, and therapies for the coronavirus, aiding the global population to better face the common threat to our health and safety. Since then, we are delighted to see that the cross-border cooperation has taken off in strategy with frequently communication between the 2 teams. The leading research teams from GIRH has been able to share on medical cooperations, its first-hand findings and experience in epidemic control and patient care and the initial 30 research projects. Today, MassCPR is announcing the funding of 62 scientific research projects, many of which are joint projects carried out by Sino-US scientist research teams. Coronavirus pandemic is a common challenge faced by the entire global world, and working together is the most effective way to win the battle. We are confident that Chinese and American research communities will taking the opportunity to further promote it and set an example for international cooperation in medical and scientific research. I have no doubt that the collaboration would make extraordinary contributions to the global fight against the coronavirus. Finally, allow me to convey my best wish to a successful collaborations between Chinese American scientist communities and sincere hopes to MassCPR for speed discoveries to combat the coronavirus. Thank you very much.
James, thank you very much. And I apologize to our viewers for some of the technical glitches here. I'll turn it back over to David Golan.
Thank you very much, George. And thank you very much for your update and for your partnership, Dr. Zhong and Mr. Shah. My name is David Golan, and I am the Dean for Research Operations and Global Programs at Harvard Medical School and a member of the MassCPR leadership team. Scientists and clinicians around the world are rising to meet the challenges presented by the pandemic. We are seeing this call to service play out every day within our Massachusetts biomedical research community. Through MassCPR, we are providing the environment and resources needed to harness the talent and commitment of our researchers in a collaborative, coordinated, and multidisciplinary approach to battling this new disease. In that spirit, I am greatly honored to announce MassCPR funding awards totaling over $16.5 million to support 62 research projects that address the most pressing challenges of the pandemic. The projects are headed by principal investigators from 15 different institutions in Massachusetts. They were selected from among some 450 applications for their potential to influence clinical outcomes in patients and populations within the next year. You can see the full list on our website at masscpr.hms.harvard.edu. These projects are organized around the consortium's key focus areas: the development of vaccines, therapies, and diagnostic tools, clinical management and outcomes research, epidemiology, and understanding how SARS-CoV-2 causes disease. We are very grateful for the financial support provided for this first round of awards by the China Evergrande Group, Mark and Lisa Schwartz, the Massachusetts Life Sciences Center, the Bertarelli Foundation, Howard Cox, Leonard Schaefer, the Abundance Fund, Jane and David Ott, and other generous donors. We continue to engage with philanthropic organizations and anticipate that additional proposals will be green-lighted as more MassCPR funds become available. It is now my pleasure to introduce MassCPR faculty co-leaders Arlene Sharpe and Bruce Walker. Arlene is chair of the Department of Immunology at Harvard Medical School and co-director of the Evergrande Center for Immunologic Diseases at HMS and Brigham and Women's Hospital. Bruce is director of the Ragon Institute of MGH, MIT, and Harvard. Arlene and Bruce.
Thank you, David. You may be wondering how we organize our work to address a pandemic of this magnitude. After the initial outreach from our colleagues at Evergrande and the GIRH to us at the end of January, George called a group of us together to deeply but quickly consider how to best mobilize our community in response to this crisis. We brought together the Boston community in March with our colleagues in China. Now we have this incredible collaboration, MassCPR. How did we do this? By establishing working groups with collaborating institutions, members from across all of Boston, as well as in China. We have established 6 working groups on the following areas: clinical disease management, pathogenesis, or how the virus affects the body to cause disease, as well as diagnostics, epidemiology, therapeutics, and vaccines. Each one of these areas focuses on a critical facet of the virus, the disease that it causes, and the challenge that it poses. As a result, we organized these 6 working groups, as not only did each area represent a major theme on which the world would have to focus to turn the tide against this pandemic, because each of them drew upon the incredible talents of faculty throughout the Boston biomedical research and healthcare ecosystem, as well as our colleagues in China. The to-do list for our working groups includes the following. On the front of epidemiology, improve disease surveillance and optimize disease forecasting models. On the pathophysiology front, unravel the biology and structure of the virus itself, as well as the disease that it causes. This includes understanding how the virus behaves in the body, how it interacts with various cells, tissues, and organs, how it causes the most dreadful complications of the disease. On the diagnostic front, design and deploy novel testing modalities, tests that detect active infections as well as past infections. We want to identify and deploy biomarkers that can help clinicians monitor disease progression and also can help forecast who may be at risk for developing critical complications. For therapeutics, develop new therapies on several fronts, from antibody-based treatments, including lab-designed antibodies, to the design of new antiviral drugs and the repurposing of existing agents. And of course, our ultimate quest is to develop a vaccine.
And thanks, Arlene, and I'll continue now just to begin to introduce the speakers. I want to stress that our immediate focus is trying to turn the tide on the COVID epidemic, but we've really come together for the long term to be ready for the next pathogen that emerges so that we can immediately respond. We're going to need to focus on the development of viral drugs and vaccines that work against multiple different viral strains. And the tools that we're going to develop are going to be absolutely critical so that we have an arsenal that we can deploy as soon as new pathogens emerge. Our plans in the MassCPR are ambitious, but we really feel that we're up to the task. We have an incredible group of investigators here in Massachusetts and our collaborators in China. I think we all went into science and medicine to transform human health, to alleviate suffering caused by disease. And this historic pandemic has really been a defining moment for us. It's a call to duty, and we're there to respond. So it gives me great pleasure to introduce to you our speakers this morning. They represent our 6 working groups, and they'll give you a window into their work, including their progress, challenges that they're trying to overcome, and summaries of key projects underway. I want to stress first that the work presented today is mostly unpublished. It's not been peer reviewed, contains preliminary data. The meeting's being recorded and will be posted on our website for public viewing. The speakers in order of appearance are Lindsey Baden from the Brigham and Women's Hospital, who's going to speak on clinical management, followed by Galit Alter at the Ragon Institute, David Walt from the Brigham and the Weiss, Megan Murray from Harvard T.H. Chan School of Public Health on Epidemiology, Jonathan Abraham from HMS and the Brigham on Therapeutics, and Dan Barouch finally from the Beth Israel Deaconess and the Ragon Institute on Vaccines. So with that, Lindsey, you have the floor.
Thank you. Thank you, Dr. Zhong, Dean Daley, Arlene, Bruce, for your leadership in bringing us together and in charting a very important and urgent course. As we all became aware in January, February of the emergence of this pathogen and the severity of illness, we in early March brought the MassCPR together, and in doing that, realized the urgency of the severe illness that this disease was causing and the need to develop ways to treat this, given that at that time there was no known effective treatment. Next slide. The clinical management group, in seeing this urgency in early March, had to determine our priorities, and one of our priorities was to develop treatments immediately, given the urgency of illness. At the same time, the MassCPR is developing longer efforts to understand the disease and develop longer-term treatment. In doing this, we set up cross-disciplinary educational conferences, developed guidelines, and urgently established clinical trials. In the educational conferences, we were very fortunate to be able to work with Dr. Zhong's group in Guangzhou and be able to learn from their experiences in treating patients, diagnosing the illness, and defining the clinical syndromes and the nature of the illness to prepare for our own response locally. Next slide. The clinical manage committee or group rapidly engaged all 4 medical schools in the Commonwealth, as well as the major medical centers, both to learn from each other and from our colleagues in China, as well as to develop treatment strategies for the patients we're anticipating seeing. And what's very important is to remember the timeline. This is March, 2 months ago. So all that has occurred has been over the last 2 months, including anticipation of the arrival of the illness and the potential management of the patients. In doing this, we developed care guidance for our different medical centers. In developing this guidance on very, very limited evidence, we also informed national guidelines through the IDSA, the Infectious Disease Society of America, as well as DHHS and NIH, as we all were preparing for the emergence of this severe illness and how best to treat it, and to iteratively learn on how to treat it better and to learn from each other. Next slide. Over February, March, and April, working together both locally as well as internationally, the illness associated with SARS-CoV-2 or COVID-19 disease was defined or has been partially defined. And in defining this, it allows us to better understand targets where we can intercede on treatment. And the illness has a viral infection phase with injury associated with direct viral replication. And what has emerged in March and April is an understanding that there is an immune diathesis, an immune dysregulation, an inflammatory state, and that inflammatory injury is also part of the disease pathogenesis that allows us to think about treatments and the timing of treatments in different phases of illness that can be against the virus or against the immune system that is causing some of the pathogenesis. This work in part is interdigitated with the pathogenesis group and Dr. Alter's work and others in defining these stages of illness. Next slide. What we urgently set out to do in late March and early April, realizing there was no known treatment that had been established to treat SARS-CoV-2 or its associated illness, was to look at agents that were on the shelf, either in clinical use or potentially for clinical use under study in other contexts, and to rapidly engage our broad Boston and Massachusetts medical community to design high-quality trials that could be deployed immediately and to develop knowledge that could inform us as to what works, what doesn't work, and what can be used not only locally, but globally. We thought of this in the categories of disease pathogenesis, agents that attack the virus, agents that interfere with the inflammatory response, and other types of agents that address some of the complications. The INO is inhaled nitric oxide, which can affect the respiratory or ventilatory challenges, the thrombotic diathesis or the clotting that occurs, maybe anticoagulation, and then other types of treatment that might be of benefit but needed rigorous study. What I have highlighted, the 4 in bold, are 4 examples that I will go over of studies that were launched. What's important to note is over the last 6 weeks, over a dozen high-quality trials have been launched across our institutions with over 400 patients enrolled who have received different types of therapies in the quest to define what's most efficacious. Next slide. In discussing 4 examples that have launched over the last 6 weeks, some of these are investigator-initiated, some are single-center, most are multi-center, some are industry collaborations, federal and other sponsor collaborations. All are immediately applicable to determine if we can find efficacious therapy. Remdesivir are studies engaged with Gilead as well as the NIH. Libby Homan led the NIH ACT study locally. These data have been reported. That study was enrolled, completed. DSMB stopped the study early a few weeks ago. An EUA was filed with the FDA to allow expanded access, given evidence of efficacy. Hopefully the data will be more fully in the public domain shortly. But this is incredibly encouraging in my view, in that in a matter of weeks, therapies have been identified that have evidence of efficacy. The Gilead studies looking at severe and moderate illness, led at some of our institutions by Katie Stevenson, Francisco Marti, at different institutions across our consortium, are providing more evidence on how to optimize treatment. And these data as well, hopefully will be available shortly, so that we now have a therapy that has some evidence of activity that we can deploy for our patients. Next slide. Another agent that was identified by members of our consortium that had clinical evidence of activity against SARS-CoV is used in Japan for influenza, but not available in the US. Engagement with the sponsor, Fuji, was done. They were interested in a collaborative study with Bob Finberg at UMass, Boris Ulig at MGH, Nick Issa at Brigham and Women's, designed, developed, and have initiated this study, and it is well into enrollment to allow us to determine if this agent has meaningful activity against this virus. I think it is quite remarkable how quickly these studies were designed and initiated. However, we need to learn how to do this even faster. This is faster than I'm aware of it being done previously, but we still are not fast enough in developing new therapies. However, these types of studies are high quality and will provide high quality directive information as to how we can treat this virus. Next slide. Tocilizumab is a biologic that blocks IL-6. IL-6 is a cytokine that is part of the inflammatory response. This speaks to the comment I made earlier about later effects from the virus with an exuberant or an overacting immune response. Given that observation, can it be targeted to ameliorate the severe immune response that can be deleterious? Dr. Stone, a rheumatologist at MGH, with colleagues across 4 institutions in the state, designed this study. Genentech became involved. This is an investigator-initiated multi-center trial that is high quality, that is conceived, launched, and is well into enrollment in a matter of weeks, and hopefully will provide us insight as to how we can treat this part of the illness in a salutary manner. Next slide. And lastly, I wanted to highlight the ESCAPE study, which is based on the concept that if you acquire SARS-CoV and you recover, your adaptive or immune response has controlled the virus and allowed you to recover. Can this immune response provide insight for future treatments? To Jonathan Abramson and the therapeutics group are working in ways to develop these types of more targeted antibodies. But can we use plasma from individuals who have recovered as a treatment? But can we do it even smarter than that format? And that's where colleagues at Beth Israel Deaconess and at the Needle at BU, where we can measure the immune response, particularly high titer neutralizing antibody to this virus, and utilize individuals who have a particularly strong immune response as a way to target who may be the best donors to provide therapy to patients who are sick with this pathogen, and we can then provide them the most targeted therapy in this format. This, I think, is one of the best examples of what MassCPR can do, which is using our science to identify high-quality therapies, leveraging our network, as this study will encompass a half-dozen centers across the state, to provide a targeted therapy with a strong scientific rationale, all developed in a matter of weeks, leveraging the significant biologic insight and technologies our community has. And I think this is yet another example of the best of what we can do as a community rapidly. So I would like to stop there, having shared these examples of different clinical management strategies and high quality clinical trials that have emerged over a mere weeks. And now turn this over to Dr. Alter, who will discuss the pathogenesis advances.
Thank you, Dr. Baden. So on behalf of my co-chair, Dr. David Knight, and the 185 members of the Pathogenesis Working Group, I'd like to take you all on a whirlwind tour of how this community has come together and to share some of the extraordinary work that they have done and plan to do. Next slide. To start, pathogenesis is simply the science of understanding the development of a disease, from the identification of the causative agent all the way to understanding the disease manifestation. As a science, pathogenesis bleeds seamlessly into epidemiology. Why do some populations get sicker? Therapeutics. Why do some medicines work on some patients and not others? Vaccine development. How can we induce responses that will mimic natural immunity? Thus, the Pathogenesis Working Group has the fortunate opportunity to work with all working groups within the MassCPR. Next slide. So the mission of the Pathogenesis Working Group is simple. We aim to determine how the SARS-CoV-2 virus causes the disease and how the body fights this infection. Several priority questions have been defined, including understanding how the virus infects, how it spreads, how the virus is sensed, why age influences disease course, how comorbidities alter response to the virus, and how we can identify protective immune responses to guide vaccines and therapeutics. Next slide. These questions launched the Pathogenesis Working Group on a whirlwind of activities that all began in December 2019. A new virus caused some rumblings in the virology community at the start of the year, rumblings that got louder once the global outbreak began to spread in early January. By early March, the formation of the MassCPR solidified the need for a pathogenesis working group that would bring together scientists and physicians to begin to think about this important question. The community rapidly assembled, reagents were shared, human samples were collected, animal models were established, and weekly technology talks were used to assemble all of the community to discuss and begin to strategize on how we can understand and move development of therapeutics and vaccines along. Next slide. Probably most inspiring is the remarkable redeployment of the scientific community across Massachusetts. Scientists from multi-institutions took a stand in early March, applying their remarkably diverse expertise to tackle this global problem, to dissect this disease, and to contribute new knowledge to the globe to help bring an end to this pandemic. Next slide. The pathogenesis community brings together scientists and physicians from biology, virology, immunology, genetics, and many more in the top right. Mathematicians, physicists, engineers in the top left, as well as multiple clinical expertise ranging from oncology, cardiology, infectious disease. The community truly represents the very best and the very brightest looking now at this disease. Each group looking at COVID-19 through a different lens, these different perspectives, and the blending of this unique thinking is what catalyzes innovation and the development of novel approaches to tackle this devastating pathogen. Next slide. The community has assembled around weekly tech talks that occur each Wednesday morning at 10 a.m. Since the beginning of April, the meeting has brought together a steady stream of 100 investigators and laboratories from across the state. The numbers are shown in the bar graph. From across these disciplines and institutions, all investigators are eager to learn about novel cutting-edge technologies. The talks foster open communication, bring together working groups, and have catalyzed the formation of dozens of collaborations that may never have happened without the MassCPR. On slide 8, on the next slide, the major challenges that the community faced can be broken down into two problems. The first, there was a critical need to begin to define expertise around the host-pathogen interaction. The community began to rapidly identify the scientists who could look at the virus, the cells that fight the virus, the organs in which the virus persists, the animal models that allow us to understand the disease, as well as to apply tools to understanding what is happening within those who are infected, and ultimately how this works at a population level. The second challenge was to try to identify the technologies that could be rapidly deployed to collect the most informative data. Focus on collecting data on the littlest molecules on the left, through to the genetic code, to genetic programs, to the protein building blocks, to the entire immune response, to understanding the biology and organs, and ultimately in deciding how we can collect data from the actual individuals that could help us think about how we can fight the disease. From these discussions, dozens of projects bloomed, and I'll just now name a few. On the next slide, the first is a truly remarkable story about a platform that has been developed by Dr. Elke Mühlberger, a virologist at the Boston University National Emerging Infectious Disease Laboratories. Elke has systematically redeployed her entire laboratory to support the pathogenesis work of the whole MassCPR. This generosity is extraordinary, allowing any investigator from across Massachusetts to partner and study the coronavirus in its natural state. Elke has developed a number of collaborations, at least 10 of which are occurring across institutions, and a few of the questions that she aims to tackle are listed at the bottom of the slide, including questions about the application of novel therapeutics and understanding what cells and how the virus infects different tissue types. On the next slide is another collaboration that has now emerged from two outstanding investigators in the consortium, one from the Broad Harvard Medical System, Dr. Pardis Sabeti, and another investigator, Dr. Jeremy Lubin from the University of Massachusetts. This team aims to define the influence of genetic mutations in the virus on their ability to accelerate disease activity. As has been reported recently, small changes in the virus have been noted at the genetic level across the globe, shown in the top left of the slide. These genetic mutations have been linked to disease severity. Now, these two investigators have partnered, bringing together ultra-deep genetic sequencing capabilities and virological expertise to rapidly link genetic alterations that are occurring in real time to disease mechanisms. Finally, the last project I'd like to highlight is another one focused here now on understanding the immune response to the virus. Now, brought together by multiple investigators across Massachusetts General Hospital and the Broad Institute, this group of investigators, with pictures shown at the bottom of the slide, have now begun to collect samples incredibly rapidly from 200 hospitalized COVID-19 volunteers. This group of investigators have redirected their research to begin to track the unique changes in cellular frequencies, cellular activation, signaling differences, and genetic changes within these individuals' precious samples to begin to understand the immunological mechanisms that lead to control and clearance of the infection. But what is most unique about this particular collaboration is the will of this group to create knowledge for the world. All the data will be posted publicly for global access, allowing rapid analysis of data from anybody around the world to accelerate innovation and interventions. In a very short time, this consortium has already generated 3 high-impact papers, and that is just the start of this enormous productivity. Finally, on my last slide, I just would like to emphasize that as the world begins to reemerge, this assembly of the scientific community has now allowed us to begin to think about how we can make those groundbreaking discoveries that will help us see the end to this particular pandemic. And this is only possible under the umbrella of the MassCPR. And now I will pass it over to the next presenter, Dr. David Walt.
Thank you, Galit. I'd like to welcome everyone. The Diagnostics Working Group is co-chaired by myself, as well as Pardis Sabeti and Bronwyn McGinnis from the Broad Institute. Next slide, please. Our mission is to ensure that everyone in Massachusetts and the world has access to timely and reliable COVID-19 diagnostics. And we have two priorities: increase access to testing locally and globally, and also leveraging the Massachusetts CPR innovation technologies to improve assays. We have a unique life sciences and biotech ecosystem here in Boston and Cambridge, including the public and private sectors, as well as the rich community of academic institutions. And these, in partnership, can be leveraged to address this challenge. Next slide. We have built a large community of working group members with over 150 MassCPR members, including weekly meetings with clinicians, academic scientists, and many other investigators. As Dean Daley mentioned, we need to learn from our colleagues' experience in China and the rest of the world. Our working group engages frequently with the Guangzhou Institute of Respiratory Health to help us accelerate our efforts and to share our mutual learnings. When we first experienced the initial cases in Boston and Massachusetts, we were unprepared for the testing demands required to meet the needs of patients and the frontline healthcare workers. And we mobilized a network of diagnostics experts and clinical laboratory leaders to accelerate the development of tests. Boston hospitals, I'm proud to say, were among the first hospitals in the country to have diagnostic testing on site. At the Broad Institute, repurposed laboratory space received FDA approval within a week and had tested 500 patients within the first week of operation. And they are now at the capacity of being able to measure thousands of tests per day. There was an incredibly rapid transformation that enabled the Commonwealth to meet the urgent needs of patients in terms of getting tested. Next slide, please. Okay. The next phase of emerging diagnostics technologies include point-of-care systems, such as will be deployed at urgent care centers, physicians' offices, community hospitals, and also the direct-to-consumer tests that will be used for home and at-work testing. This distributed type of testing, we believe, will be important to getting all of us back to work and back to the new normal. Many of these testing platforms, as we've heard, are of marginal quality. At the Mass General Brigham Center for COVID Innovation, supported through a grant from MassCPR, we have set up a test bed called the Diagnostics Accelerator, where we can test and validate these platforms so that they can be rolled out into the community with the confidence that they will be delivering accurate results. Next slide, please. COVID samples are precious. In an unprecedented demonstration of collaboration and cooperation, we have been able to set up a COVID sample bank where all MassCPR institutions make their samples available to researchers at all the member institutions. We have participation from a growing network of institutions, as you can see, including Tufts Medical Center, Brigham and Mass General, Beth Israel, Boston Medical Center, and this is growing through additional negotiations with other institutions in Boston and the greater Massachusetts area. We also hope to extend sample access to local companies pursuing COVID research. Next slide, please. Our efforts are focused on accelerating the next generation of technologies that will address both near-term and long-term testing needs. These will include such areas as massive multiplexing, where we can pool a large number of samples and use advanced technologies such as next-generation sequencing to literally measure tens of thousands of samples at a time, which will be critical for population testing. we have supported through MassCPR point of care technologies, such as those of Wesley Wong at Boston Children's and the Wyss Institute, who's working on DNA nanoswitches. Feng Zhang at MIT and the Broad Institute, working on CRISPR-based diagnostics. And Michael Springer from Harvard Medical School, working on rapid and cheap sensitive assays. Also, Lee Gerke from MIT is working on rapid tests to enable home testing. We also are interested in looking at exposure testing, and we'll hear soon from Megan Murray talking about epidemiology. These kinds of tests will be critical to being able to understand how infection and the prevalence of infection penetrate into our society. George Murphy from Boston University is developing large-scale screening methods to better model infection in the population. Dwayne Weissman is observing the human antibody response to the virus to predict infection risk. And Steve Elledge is applying his VirScan to comprehensively scan the immune system of both coronavirus as well as other pathogenic organisms. Next slide, please. We still have a lot of work to do. We need more testing. We need continued innovation. We have to have faster, more distributed, more sensitive, and more comprehensive testing. But we also need to use this testing to solve problems that are broader. How can diagnostics make a difference? Despite this focus on innovation, we are focused on the ultimate implementation of these diagnostics and testing technologies to help our city, state, country, and the world safely get back to work, school, and our new normal. We hope our work will set an example for how to tackle this problem comprehensively and for us to be prepared for the next pandemic. These are the true goals of our work. Thank you for your attention. Now I'd like to introduce my colleague, Dr. Megan Murray from the Harvard T.H. Chan School of Public Health. Megan?
Thank you, David. So I am the co-lead with Michael Minna of the Epidemiology Working Group. And next slide, please. Our mission is to understand the local and global distribution, impact, and mitigation of emerging pathogens in general, and of course of COVID-19 specifically. So among the many priorities that we could ascribe to epidemiology, in this particular case, we want to measure the extent of COVID-19 locally and globally, to model the impact of interventions, to identify determinants of disease and disease outcomes, and to measure the efficacy of prevention measures. Next slide, please. So as a study of emergence, as a study of emergence, distribution, and control of disease, disability, and death among groups of people, epidemiology covers a lot of ground in general, and for COVID-19 in particular. There were so many aspects of the epidemic that fall under the heading of epidemiology that it's been challenging to keep up with all the areas and the many people in our community who work in this area. But let me highlight some of the areas in which people in our community are working. We have a very strong community here at the Harvard Chan School, at MIT, and within the hospitals that include some of the world's leaders in the area of mathematical modeling of epidemics. I want to highlight some of the work focusing on model impact on mobility, social distancing interventions, the potential impact of seasonal variation in COVID transmission. We're also focused on applying some of these modeling techniques to understanding what is happening in other countries, especially in Africa and parts of Asia, where the epidemic is late to arrive. A great deal of work is underway to address how best to measure the burden of COVID-19, both in terms of how many people have been infected and so now may be immune, and how many people are currently infected, how many people will require hospitalization, ICU, and ventilator use, and how that aligns with the current availability of these tools. A large group is focused on identifying pathogen and host variation and thinking about how that might account for different outcomes on infection, both at the population level, why are some countries relatively spared and others not, and also on an individual level, why do some people have mild or undetectable disease while others progress rapidly to acute respiratory distress syndrome? And finally, since EPI is not just the study of an epidemic pathogen, our working group includes those looking at other outcomes of the pandemic, whether those are impacts on our economic health, the health of societies in general, and in particular, the mental health of those experiencing the epidemic. So I wanted, next slide, please. Let me highlight a couple of the projects that are underway. So Michael Minna is a physician and epidemiologist who is working to understand the burden of infection and disease through a series of seroprevalence and viral assessment studies in populations in Massachusetts and elsewhere. He's looking at healthcare workers, first responders, nursing home residents, among many others. He and his colleagues, we've heard from Galit Alter, who is involved in this project, have set up the Immune Observatory pilot program, which is about to kick off and involves a collaboration with Octapharma to continuously monitor 100 selected sites across the US for the prevalence of SARS-CoV-2 antibodies. They'll use discarded specimens obtained from 500 plasma donors weekly from each site. The sites are carefully chosen to offer a representative sample across the country. They'll be shipped to the Immune Observatory for measurement of SARS-CoV antibodies using a high-quality quantitative SARS-CoV receptor binding domain ELISA assay that's been developed by the MassCPR and lead Galit Alter. Next slide, please. In another project that's designed to look at to assess the burden of infection locally, Timothy Erickson is leading a project that uses a very different method of estimating the levels of infection in different communities. He works with a group of researchers at MIT and the company BioBot Analytics to sample wastewater. We now know that SARS-CoV-2 is shed in stool, and that means that it's can be collected in sewer systems. Tim and his colleagues are using this to do what they call wastewater epidemiology, to rapidly gauge the level of COVID-19 infection in a broad community. This helps obviate the need to identify and sample specific individuals and to deal with questions like whether or not to return results to them, which was especially problematic before some of the tests had been approved. The team plans to enlist communities around the country to sample their own wastewater and ship samples to Boston for analysis, thereby creating a real-time map of the communities and how they are affected over a given period of time. In another project, next slide, please. I'm sorry. In another project, a team at Harvard Medical School led by Bethany Het-Gautier has been asking similar questions about the extent of infection, but focusing on low-income countries where COVID-19 cases are believed to be very much underreported. Since diagnostic tools aren't available in many of these countries, we can't use the kinds of methods we've been looking at that Michael and Tim have been developing. Bethany and Izzy Fulcher, her postdoc, have been looking at routinely collected health management and information data that's publicly available from multiple countries throughout the world. They focused on 7 countries to see if they can detect a pattern of a syndromic surveillance signal that would indicate an unexpected increase in the number of acute respiratory infections that have been reported over the last few months. This involves setting a baseline for what would be expected in this period, looking at other years, and then managing ways to accommodate the high levels of missing data. What you see here is data from a single county in Liberia, where the red indicates what has been observed in the past, or expected in the past based on previous year's data. And then the black shows a spike in January, February, and March of this year in what has actually been observed. Next slide, please. David Reich leads a very productive and prestigious lab that works on the genetics of ancient humans and uses genomic methods to track migrations of ancient people But in this case, his team has redeployed and is using similar genomic methods to understand and track COVID-19 as it evolves over time and space. Harold Ringbauer is a postdoc working in the Reich Lab who has used the publicly available SARS-CoV-2 genomes to track the progression of a set of 4 very common mutations that always appear together in the viral genome. This strain, which has these 4 mutations, is now accounts for about 65% of the observed genome throughout the world and is rising in almost all globally sequenced cases. Harold speculates that the rise in this strain of COVID-2 is probably not due to selection, but rather to a bottleneck, meaning that it just got lucky early on. But the team is doing work that really aligns nicely with the work you heard about from Pardis Sabeti's group, that is looking to see whether these genetic changes might be associated with phenotypic changes that would change some aspect of the virus. I think this question of whether these mutations lead to changes in transmissibility is a very important one. And genetics can be used to track this kind of epidemiological patterns across the world. Next slide, please. This is work that my group is doing. We are very interested in conducting a clinical trial of BCG vaccination to see if it has efficacy against COVID-19. So BCG, Bacille Calmette-Guerin, is a 100-year-old vaccine that was used against tuberculosis since the 1920s. It's an attenuated strain of a mycobacteria that's closely related to the bacillus that causes tuberculosis. And it's been an effective vaccine for children who have TB. But it's also been shown, somewhat surprisingly, to have a non-specific effect on child mortality that is not mediated through tuberculosis. So it appears that BCG has been modulated immune systems so that children are less likely to die of diarrheal and respiratory diseases if they've been vaccinated. More recently, researchers have shown that BCG can alter innate immune cells through epigenetic modification of their genetic material. This might mean that BCG could have an impact on the immune response, the innate immune response to COVID-19. And we and other groups are working to run clinical trials to see if that effect is real. So this conglomeration of very different projects has really blossomed under the organization of the MassCPR, bringing together lots of different people who can think through how best to approach these problems and use the resources available in this amazing community to address them. So thank you.
I'll be happy to take over now. So I co-lead the Therapeutics Working Group with Dr. Mark Namchuk. Next slide, please. And the mission of the Therapeutics Working Group is to meet an urgent need for drugs to prevent or treat infection by emerging pathogens. And we've subdivided this into 3 priorities. The first one involves repurposing currently available drugs. The second one is to screen small molecule libraries to develop new drugs. And the last one is to develop therapeutic antibodies or biologics. Next slide, please. And so by repurposing, what we mean is to rapidly determine if existing drugs can be used to treat COVID-19. And so here on the top part of this slide, also showing you data here from Rob Davies' lab at the National Emerging Infectious Diseases Laboratory at BU. What we have here are uninfected cells shown in blue. Those cells can be exposed to a drug. A virus is then added, in this case, SARS-CoV-2. And after an incubation period, ultimately, Rob's team is able to detect infected cells by staining these cells with antibodies against viral proteins being made in those cells so that the cells effectively glow green. And what you'll see at the bottom left of the slide is a robotic drug aliquoting system that Rob has been able to set up, where it's set up with 384 different drugs can be tested simultaneously to look for an effect on viral infection. And so if one were then to stare down these wells using a microscope, and these are images I'm showing you at the bottom right, the bottom left image, you'll see an example here where there are blue dots representing individual cells. In this case, the drug perhaps is killing cells since you don't see a lot of blue dots, and this is an outcome we would want to avoid. In the middle panel here, you'll see a situation in which the drug has no effect. So you see a lot of blue dots, but also a lot of green spots, suggesting again that the drug has not been able to block viral infection. And then if we were to look down the wall and see what we see at the bottom right here, we'd be quite excited because there we have a lot of blue cells, so blue nuclei, but not a lot of green, suggesting that we found a drug that blocks virus infection. And so Rob is able to do this for thousands of drugs. And the advantage of repurposing is that we're now using drugs, in some cases, that are already used to treat other diseases. And so we know that they can be safely taken by patients. And we also know what side effects to look out for. So we think of this perhaps as the most promising strategy to rapidly have an impact on the ongoing COVID-19 pandemic, because again, those drugs we're more familiar with. Next slide, please. And so a more traditional approach to discover drugs against viruses involves targeting their viral enzymes. So these are the workhorses the virus uses to productively infect cells. And so examples of where this has garnered great success include HIV-1, hepatitis C, and influenza virus. And so one effort, for example, by Dr. Mark Namchuk being led at HMS is targeting the viral protease. So this protein here, I'm showing you the molecular structure, is an enzyme that is required to cleave other proteins the virus produces in order to activate them. So if you block this enzyme, you block virus replication and presumably you'd be able to develop an effective drug. Now, traditional efforts targeting viral enzymes involve using small molecule screens and compounds, and this can take some time on the scale of years. But I want to show you on the next slide an approach that we're also quite excited about that's being used by Dr. Hari Artarnari at Dana-Farber Cancer Institute and Harvard Medical School, where he's partnered up with Google and computer engineers there to use supercomputers to find drugs. And so what Hari's been able to do is test billions of compounds in a few weeks instead of several years. And here I'm showing you examples of some of his early results. So on the left here is a SARS-CoV-2 polymerase. So this is the protein the virus uses to copy its genetic material. So you can think about it as a copying machine. And so the active site, or the business end of this molecule, is shown here where the compound A is binding. And this site is exactly where remdesivir bound, the drug we heard earlier about by Lindsey Baden, has shown some signs of promise for clinical use in humans based on early clinical trials data. But perhaps Hari's molecule could be a more favorable molecule to be used. It might be more potent, but this is an exciting lead that Hari's now following up on. And the bottom right, I'm giving you an example of the SARS-CoV-2 main protease, the enzyme I showed you on the last slide, and a compound here bound in its active site that Hari's also following up on. And so through resources made available through MassCPR, Hari is then taking these compounds, sending them to the Broad for them to be further processed, and then ultimately sent to the needle where Rob Davies' team is going to test them against live virus using some of the infectivity assays I showed you on the previous slides. Next slide, please. And so you've also heard about antibody therapy from what Lindsey had mentioned earlier. So here I'm showing you a virus, and these red blobs on the virus surface are called the spike protein. And what you're seeing floating around the slide are these Y-shaped molecules called antibodies. So these are the molecules our immune system makes to fight off virus infections when we're infected by a given virus. Next slide, please. And so where antibodies can intervene, so I'm showing you here a picture of a small virus with spike protein on its surface. We know that SARS-CoV-2, to bind the cell, binds a molecule called ACE2. After ACE2 binding, the virus is then internalized to the cell, and there's a last step that the spike mediates, whereby the virus has to penetrate through the membrane of the cell in order to dump its genetic content and proteins here, which I'm showing you as squiggly lines inside the viral particle. And this is really what kicks off the infectious cycle. And so antibodies can intervene at several steps, including first blocking attachment to the ACE2 receptor, or downstream of this, preventing the membrane penetration step by locking the spike and preventing it from carrying out this membrane penetration step. Next slide, please. And so we're lucky to have, as part of a consortium, Dr. Wayne Marasco. So Dr. Wayne Marasco has previously had experience developing antibody-based therapeutics for SARS-CoV as well as MERS, so two related coronaviruses that have caused smaller outbreaks in the past. The approach Wayne has been using involves taking blood from healthy human donors, which he did this for 57 donors back in 1996. From these donors, he was able to get the immune cells, extract from them the sequence that includes various antibodies found in their blood. He's able to present these onto the surface of a microbe here. These are bacteriophages shown in the middle of the slide. So each bacteriophage here has an antibody on its surface, and there are 27 billion different combinations. He's able to amplify these in the lab by growing them in flasks. He's able to test which microbe can bind the spike protein, and then from the microbe can backtrack exactly what antibody was on its surface that gave it that activity. So Wayne has rapidly used this to identify over 70 antibodies that target the viral spike protein, and I'm showing you some examples here of some antibodies he's shown to be quite potent at neutralizing the virus using the SARS-CoV-2 infectivity assay with Dr. Anthony Griffiths at the Needle. Next slide, please. So now a number of labs in the consortium, including investigators at Boston Children's Hospital, Brigham Women's Hospital, MGH Ragon, and including my lab, has been working hard as well to identify antibodies from the blood of convalescent donors. And so the way this is done is we, again, through a resource made available through MassCPR, can take blood from donors that have recovered from infection several weeks prior. We can isolate the immune cells, which I'm showing you here in the middle of the diagram, the B cells that make antibodies on their surface, label them with a fluorescent spike protein we've also engineered in the lab. And what you'll see at the rightmost part of the slide is the final flow cytometry plot. So this is an instrument that allows us to look at, in this red rectangle here, each little dot is an individual immune B cell that is effectively making on its surface an antibody molecule that can bind the spike. And so from these cells, we can extract a DNA sequence for antibodies. And my lab, for example, has sequenced more than 80 antibodies thus far that effectively are reacting against the spike protein. And so next slide, please. Ultimately, our goal is to rapidly put molecules to the test. So I've mentioned several times the partnership with the National Emerging Infectious Diseases Laboratory at the Needle, which has been critical to our ability to rapidly move therapeutics towards human use. The next step they're getting ready for now is preclinical efficacy testing, which we're quite excited about. And now I'll be happy to pass on to Dr. Dan Barouch from BIDMC, who will be telling us about work done on vaccine design.
Thank you very much. What I will do is I will discuss some of the work conducted in the MassCPR Vaccines Working Group. Next slide, please. So our mission is to develop effective vaccines to end pandemics. MassCPR is not restricted to COVID-19, but obviously is the primary focus for now. So the priorities are to develop effective COVID-19 vaccines, to advance rapid vaccine development for pandemic control, and to contribute to vaccine science. Next slide. So the first question is, why do we need a COVID-19 vaccine? The explosive spread of this epidemic, as well as the spread from asymptomatic persons, suggests that a vaccine may actually be required as the key tool to end this pandemic. In addition, in case the natural trajectory of the epidemic is downwards, then there's a likelihood that the COVID-19 will reemerge and cause future outbreaks, maybe even seasonal outbreaks, and a COVID-19 vaccine could be important to control those future outbreaks. And regardless, the exploration of protective immunity and immune correlates of protection will be an important contribution to science. Next slide. We are cautiously optimistic that the development of a COVID-19 vaccine is technically feasible. This is based on some data, but I should emphasize that there's a lot that's currently unknown. Unlike other pathogens such as HIV, it's clear that most people who get COVID-19 recover. So the human immune system appears to be able to contend with and eliminate this virus. We don't yet know for sure whether those individuals who recover from COVID-19 are protected against re-challenge, but hopefully data regarding that will be available soon. In addition, there have been some mutations described in the COVID-19 virus. However, overall, the strain diversity is relatively limited. So as of now, our hope is that a single sequence will be able to be sufficient as a vaccine. In addition, previous work from SARS and MERS and other coronaviruses strongly suggests that the key antibody target is a spike protein. And therefore, we are relatively confident that we know the antigen needed in a vaccine. However, there are also technical concerns. For example, there's a possibility that there may be future mutations, and those could compromise vaccine efficacy. There's also a theoretical concern of disease enhancement. I should emphasize this has not been seen for any COVID-19 vaccine so far, not in animal models and not in humans, but it is a concern that scientists need to investigate. In addition, the timelines are incredibly fast, but are not fast enough for the current epidemic this spring and summer. Next slide. There are currently over 100 vaccine candidates under development according to the WHO. These include many different platforms: DNA, RNA, protein, vectors, and inactivated viruses. The first vaccine to reach clinical trials is an RNA vaccine from Moderna, but the timeline to initiate clinical trials and even to show clinical efficacy is actually not the same as the timeline to produce and deploy 1 billion doses worldwide, which is really the timeline that is necessary for pandemic control. If all goes well, there may be vaccines for emergency use authorization this fall or winter. And I should say that's a big if. That assumes that everything goes absolutely perfectly and goes as fast as possible. Next slide. The MassCPR Vaccine Working Group, our initial goal is to facilitate collaboration and communication among researchers working on COVID-19 vaccines. This working group is open to vaccine research laboratories in Massachusetts, really to all of them who are interested. Currently, over 40 groups participate, and we have close interactions with a MassCPR Pathogenesis working group. And as this work progresses, then there will be very close interactions with the epidemiology and the clinical working groups as well. We have biweekly meetings with informal presentations and discussion, and we're planning symposia around topics of interest. Clearly, none of these interactions could have occurred without the MassCPR. Next slide. There are multiple vaccine programs currently under investigation in the MassCPR, some clinical, some preclinical, but all scientifically informative. This is just a partial list of vaccine research occurring in the MassCPR. I can emphasize there's actually 2 programs that are aimed for phase 3 clinical trials. One is the RNA vaccine with Moderna, and my co-lead, Andrea Carfy, is leading that effort. And the second is an Ad26 vector-based vaccine with Johnson & Johnson. We are also looking at DNA vaccines, adeno-associated virus vectors, herpes simplex virus vectors, network-based T-cell vaccines, various spike and RBD proteins, as well as adjuvants, with many lead investigators shown here. Next slide. Another topic of interest is animal models for this disease and how they can inform pathogenesis as well as vaccine testing. They will also likely be required by the FDA prior to initiation of large-scale vaccine testing in humans. These are to evaluate safety as well as to exclude the possibility of disease enhancement, to look at the immunogenicity and protective efficacy of vaccines, as well as to define immune correlates protection. There are small animal models such as mice, ferrets, and hamsters, as well as large animal models such as rhesus and cynomolgus macaques that will likely be required for this effort. Next slide. So the question is how to accelerate vaccine development. Vaccine development typically takes many years or even decades. So the development of a COVID-19 vaccine in 1 year, should that effort be successful, would clearly be unprecedented in the history of vaccinology. The way this is done is to use vaccine platforms that have already been tested in humans for other pathogens. And development activities are performed in parallel rather than in series. For example, companies often are starting large-scale manufacturing before they even know if the vaccine works or not. There has to be a tolerance of a high level of programmatic and financial risk, but importantly, safety is still the most important part of any vaccine, and there should never be a compromise to patient safety in the attempt to go quickly. In addition, for vaccine development, there needs to be plans up front for mass production and distribution, because the goal is to achieve herd immunity to end the pandemic, and without mass production of the vaccines, this would not be able to be achieved. Next slide. So in summary, In the MassCPR vaccine working group, vaccine development for COVID-19 is proceeding faster than for any other pathogen in history. That's in the field as a whole, as well as the groups in the MassCPR. We believe that multiple vaccines should be pursued in parallel, since it is not clear which vaccine will ultimately prove the safest, the most protective, and ultimately the most deployable. Preclinical challenge models will likely yield data on the efficacy of these vaccines imminently, and clinical efficacy studies are scheduled to be conducted this summer and fall. Also, there are 7 billion people in this world. There has never been a vaccine program that has achieved that number of doses of a vaccine. So it would be wonderful if not one vaccine is successful, but actually if multiple vaccines are successful and reach the finish line. I believe that's my last slide. Great, and I will turn it over to Arlene Sharpe, who will introduce the next speaker, Ron Corley.
Thank you, Dan. I'd like to thank you and all of our speakers for representing the 6 working groups and for your wonderful presentations. I'm now delighted to introduce 2 speakers who will discuss what is being done to share resources to enable meaningful results. Ron Corley, who is director of the National Emerging Infectious Diseases Laboratory at Boston University, and Karen Jacobson, who is Associate Professor of Medicine at the BU School of Medicine.
Thank you. Next slide, please. You've heard a little bit about the needle and the collaborations between members of the MassCPR with faculty within the needle already. The needle is Boston University Research Institute that is devoted to emerging infectious diseases research. When the COVID-19 pandemic began, there were no faculty within the needle who were working on coronaviruses. And of the 23 investigators in the needle, almost half of them have already turned their attention to working on this virus in partnership with other members of the MassCPR. This is a remarkable turnaround because it's catalyzed in part by funding from the MassCPR. Now, being able to work with live viruses is critical to our ability to not only understand a pathogen, but to develop better diagnostics and therapeutics and vaccines. Next slide, please. Now, working with human pathogens require that you work at one of several biosafety levels. Biosafety levels 2, biosafety level 3, or biosafety level 4. This figure shows you differences in the personal protective equipment that individuals wear. In biosafety level 2, it's lab coats. In biosafety level 3, it's purified, powered air purifying respirators and garments. And then at biosafety level 4, it's a fully encapsulating suit. Each of these biosafety levels, people who work at them work in the same way. They work in biosafety cabinets, but the engineering controls as well as the personal protective equipment changes with each of these safety levels. Because SARS-CoV-2 is a biosafety level 3 pathogen, we're able to work safely with that pathogen both at biosafety level 3 and biosafety level 4, and this amplifies our ability to work with other members of the MassCPR. Next slide, please. Now, working with in containment labs, biosafety level 3 and 4 requires an enormous infrastructure and a dedicated team of engineers and facilities management, as well as environmental health and safety specialists. And in the needle, 40% of the entire building space is dedicated to support the containment laboratories. On the right side, you see some of the filtration units for filtering the air that comes out of the biosafety level 3 facilities. Next slide, please. This shows you inside a biosafety level 3 facility. The left figure is the entry sequence. These facilities have 2 interlocking doors that you have to go through before you enter the lab. The view on the right is showing you several people working in biosafety level 3 laboratories inside the needle. Next slide, please. Biosafety level 4 requires even more engineering controls. For example, there's an entire floor that has a HEPA filtration desk. These are the high efficiency particle air filtration that is required for the biosafety level 4 facilities. You have to filter the air twice coming out and filter the air once going in. And then on the right, you see what are called the cook tanks. Cook tanks are required because when you inactivate liquids in the laboratory, you put them into the piping system that goes into these EDS system that then re-sterilizes them. So it's a very high level of safety that's involved in this research. Next slide, please. This shows you inside the biosafety level 4 facilities within the needle, 2 of the lab spaces. The red hoses are connected to compressed air, which is used to pump air into the suits of the individuals working at biosafety level 4. Next slide, please. So this shows you how you get into these level 4 facilities. On the right, on the left, an individual is going into a small room. This room has 2 interlocking doors. You close this outer door and then you are able to open doors into the inside. And you can either go directly into a biosafety level 4 laboratory or into a central corridor which connects you into the biosafety level 4s. This small room also serves as a chemical shower when individuals come out of the BSL-4 laboratory. Next slide, please. So this just shows you some of the teams that are working. About half of the faculty that are working with live coronavirus work at biosafety level 4, and the other half are working at biosafety level 3. And virtually all of them are collaborating with other members of the MassCPR, which has been an incredible catalyzing event for working with this virus. Thank you. And now I will turn it over to Karen Jacobson.
Thank you, Ron. I'm going to now speak briefly about what the experience has been trying to set up at the hospital level organizations to capture patient cohorts to study and support the understanding of COVID-19 across the MassCPR. Next slide. The MassCPR has really been a platform that's allowed these 5 medical centers, all academic in nature, but serving a diversity of patients, to come together and communicate on how best to recruit individuals into prospective studies, as well as gather data and observational cohorts and consider organizations around discarded specimens. Historically, individual investigators might have reached out and collaborated, as been described in many of the exciting projects led by many of these working groups. What's unique about what we are setting up here in these sample collections and in the clinical management and outcomes data repository is to be developing platforms that will be useful for many, many projects, many not yet even developed, by having a harmonized platform where samples are sampled at time points that are coordinated across institutions, as well as thinking, how do you take very complex data and put them into a single data set that would allow for analyses across these institutions. Our institutions support a diversity of patients ranging across age ranges, racial and ethnic backgrounds, people experiencing homelessness, persons living in nursing facilities. If we remain just at one of our institutions, that opportunity to look diversely across our institutions would be lost. These platforms, particularly in the clinical management and outcomes data, also allow us an opportunity to look at smaller groups of populations, such as women who are pregnant at the time of their COVID-19 infection, persons experiencing specific immunocompromising states, including HIV, and other special populations that can be looked at only if you really are looking across these institutions. This MassCPR is really allowing some very unique and exciting platforms to be developed both for this outbreak as well as has been described, potential future pathogen investigation. Can go to the next slide, please. I'm going to focus for a moment at one institution, which is Boston Medical Center, which is the academic medical center for Boston University. This has historically been the safety net hospital for Boston and now captures and supports patients diversely in our part of the city. Just to allow people to understand the rapidity, as others have described, of how these processes have been set up. On March 13th, our institution, similar to others, had to halt almost all in-person research activity on that day, leaving many investigators having to shut down many years of work and putting into pause what have been really rich and diverse research platforms. However, simultaneous to this big push, many of us realized that we needed to simultaneously be setting up dedicated COVID research focuses and reached out across our institutions, many being physician investigator-led studies. By March 25th, this focus had already been changed, so in less than a 2-week period. And already, as described by Lindsey Baden earlier in this talk, we had at our institution 10-day turnaround of 3 trials that were ready for launch within our setting. Already now, within 2 months, we have 53 COVID research opportunities that are either active or in the pipeline, similar to activities that are happening at the academic centers across our city, many of these in cross collaboration. This has all been complex in terms of the rapidity of setting up centralized review, scientific review committees, and COVID-specific protocols, many of which have to overcome the challenges of personal protective equipment, how to consent in multiple languages without the ability to necessarily be face-to-face or using masks, and so on. Next slide. I think what we've seen during this crisis is that when you really are able to marshal the diversity of talented individuals, you can overcome many things that have historically taken months or even years to work out. And I think we've all been really blown away by how these systems can be set up so quickly. We've been able to pioneer many of the complex services that would, in other settings, take long periods of time to develop. And we now have what I think will be a very rich infrastructure across the Boston and Massachusetts platform to do very exciting translational work and understand critical nuances of the natural history of this disease that really will allow for breakthroughs to support the exciting basic and translational work that has been described this morning. Thank you.
Thanks very much both to Ron and to Karen and to all of our speakers for filling us in. I'd like now to turn to Mark Schwartz, who together with his wife Lisa has given generously to support the work of the MassCPR. Mark, would you be willing to share briefly why you chose to support this work?
Sure, thank you, Bruce. So on this call today, Lisa and I are representing all the donors and supporters of the MassCPR and the individual medical and research institutes. I'm sure I'm speaking for all the donors when I say we're blown away by what's been accomplished in just the last 10 weeks. I'd make a few quick comments touching on 3 themes. The 3 themes: 1, collaboration; 2, accountability; and 3, results. So first, as many scientists have already noted, the teamwork and collaboration has been impressive. That's been notable across many labs and institutes and many universities and hospitals in this consortium. But it's been especially encouraging to see how much sharing of ideas and data and results there's been between US and Chinese scientists. At a time when there's been little collaboration between our 2 governments, there's been a huge amount of trust and confidence at a personal level, at a lab level. So a big thank you to our friends at the Guangzhou Institute for Respiratory Health, at Tsinghua, at Baden, at Fudan, and many other Chinese universities and hospitals. The second comment, Bruce, I'd make from the donor community is how quickly the MassCPR got organized into 6 working groups and how much progress has been made already. Donors like immediate responsiveness and they like accountability, and the working groups have provided that. There's been an urgency to the work without sacrificing excellence or integrity, and that's been exceptional. The third comment, Bruce, I'd make is about results. Many donors have appreciated that there's been a very practical application of all the science and research coming out of MassCPR. Donors not only want to support building libraries of knowledge, of course that's absolutely essential, so we'll be better prepared to understand future viruses and pandemics. But we also want to resolve this crisis. We want results. So we're encouraged by the initiatives at MassCPR to do just that. Many of the efforts have been highlighted this morning, and they're all important. One, the effort to launch high-quality clinical trials in order to inform new clinical interventions. Secondly, the effort to innovate next-generation diagnostic equipment. Third, the effort to create a valuable sample biobank. Fourth, the effort to engineer better PCR and serology tests. Fifth, the effort to establish better serosurveillance systems. Sixth, the efforts to create appropriate back-to-work protocols. Seventh, the efforts to develop better therapeutics and treatment regimens. And eighth, perhaps most importantly, the efforts to develop safe and effective vaccines. As donors, we get to support all of that at MassCPR. To all of the scientists in this consortium, we thank you for your remarkable work. Thanks, Bruce.
Thank you, Mark.
Thanks so much, Mark. Thanks for your reflections. Thanks for your support. So you've heard from many of us about the work of MassCPR to stem the tide of COVID-19. Now we are eager to answer some of your questions. If you're joining us on Zoom, please post your questions using the Q&A functionality on your screen. You can direct your questions to any of our speakers today, or you can post them generally. I'll direct them to the appropriate speaker. We have the leaders of our working groups on the call. And just a reminder for members of the media, we're going to ask you to stay on this call and pose your questions during this session. We will not be having a separate press briefing. So I'll be monitoring the Q&A function and invite all of you to submit your questions. So I see our first question. It says, for antibody testing, the big question remains whether people who've tested positive for antibody are actually protected or not. How would this be determined, and what measures have been taken to determine that? I might encourage Galit Alter to address that question.
Thank you, George. So I would love to answer this question with Michael, if he'd be willing. So what I can say is that we have built really incredibly large serosurveillance studies across the MassCPR at multiple different sites with multiple different hospitals. And the idea is to start tracking folks who are antibody positive, and more importantly, to begin to understand whether or not they are truly protected. The issue right now is that we recognize that antibody testing is truly a measure of exposure. It tells you that you have seen the virus at some point, that you have developed an immune response. The only way we will know if it is actually protected is to truly begin to track these individuals and ensure that they are not being re-exposed even when they may come in contact with the virus again. That being said, in partnership with the vaccine program, with Dan and Andrea and many others, we are also learning the levels of antibodies that may be critically important for protection also in animal models. So across the MassCPR, with epidemiology, with vaccinology, with the vaccine group, as well as with the therapeutics group that are developing monoclonal therapeutics, we are rapidly learning what antibody levels are critical to really give us that immune passport that tells us that we were protected. Michael, I don't know if you're there and if you can maybe add something on the serosurveillance side. He may be gone.
And Jen might want to add something about the, you know, how animal models might be able to help with that question.
That's right.
Sure. Some of those questions can be addressed in the animal model a lot faster than they can be addressed in clinical trials, with a caveat that it is a model and those results will need to be confirmed. For example, one could evaluate very directly different titers of antibodies and whether they do or do not protect. And so we hope that there will be a lot of data in the preclinical area that will complement the clinical data to be able to answer those questions.
Great. Thank you. We have another question for Dr. Abraham. Could you speak about monoclonal antibodies and the antibody strategy compared to antiviral therapies?
Yes, thank you. That's a great question. I mean, the way I think about this strategy is perhaps a strategy that could get us to a drug quickly before vaccines are available at a later time point. So in the field of emerging viruses, it's often been explored to use, as Lindsey mentioned, convalescent plasma. But what we can do in this day and age with some of the technologies I described is use monoclonal antibodies of defined activity and specificity as cocktails and get these into patients and also scale up, for example, in the case of a number of antibodies that were developed against Ebola virus with the help of the pharmaceutical industry. And so, for example, looking at the 2014, 2016 Ebola virus outbreak and the more recent one, a number of labs identified monoclonal antibodies that were rolled out into clinical trials with some demonstrating benefit as antivirals in curing infected individuals. So the other role for antibodies could perhaps be one of prophylaxis, so to prevent infection in high-risk individuals like healthcare workers. The way to think about this again with relationship to vaccines is a passive vaccine or immunization really by transplanting the antibodies directly into individuals as a way to prevent infection or treating infection. So I think it's an important strategy to explore.
Great. Thank you, Jonathan. A question for perhaps David Walt. When do you think that individuals will have self-performing COVID-19 diagnostic tests to know their infection status?
Thanks, George. I think that's a question that everyone would like to know. As I mentioned, the Diagnostics Accelerator is evaluating some of those tests right now. They're called the lateral flow assays. These are the paper strips or plastic strips that look like pregnancy tests. And we did a horizon scan of these and found that there are over 300 commercial offerings. Most of them are from what I would say not very reputable companies or companies that are just putting things out in the hundreds and not scalable. What we've been able to do and what we're doing right now is evaluating the 10 top selected test kits that met all the criteria and specifications for being able to scale, but also having the requisite sensitivity and specificity. And we're putting them through the paces right now. But even if those work well, the issue is really one of a regulatory nature, in that the FDA historically has not allowed those kinds of tests to be introduced directly to the consumer for in-home testing or at-work testing without extensive validation and going through the full approval process, which in the shortest possible time frame would take at least 3 months. And so I would predict that we will begin to see these kinds of testing capabilities being introduced in the late summer or early fall. Most of them initially will likely be the antibody tests, but then I think some of the direct viral tests will also be available shortly thereafter. But again, one has to remember what Galit Alter just said, is that the antibody tests just tell you that you've been exposed. The hope is that by then we'll have some information about whether you're protected or not, and so the value of those will go up. But again, I think late summer or early fall would be about the right timing.
Thank you, David. I'm going to ask our Chinese colleagues to unmute and answer a general question. There is a question from Wu Wei Zhang of the paper. How do you feel about the collaboration with the Harvard teams and the MassCPR? And then also considering and about a second wave of infections in Wuhan. Dr. He, perhaps?
Yeah, past our 11 conversation and the more discussion, and some project we apply just like the use of AI based on the CT scan on the CXR and research, and I think also another like the vaccine or the experience combined together. Just like the vaccine, I really think is really important. And in China, we investigate how about in the people immunity, maybe less or 1% the people they have the immunity. So I think the vaccine is also is very important, and so many technique, and we can exchange our experience how to speed up.
Thank you, Dr. He. Yes, and indeed, I should note that we've had very productive communications about how to develop and accelerate vaccines. I will make that transition then back to Dr. Dan Barouch. There's a question from Christina Hazanetze from the Harvard Gazette, and Dr. Barouch said there may be a vaccine available in the fall or winter for emergency use. Can you speak more about how emergency might be defined, and will it depend on vaccine availability or other factors? Dan.
So thank you. So emergency use authorization is a designation by the FDA that is very different than approval. It is used only in crisis situations when there is a true need, and often that is granted based on phase 2 or early phase 3 results prior to the rigor needed for actual approval. So it's been discussed widely. It is not clear how the regulatory authorities will implement that, but it is possible. And I should emphasize I use the word may and possible with the most optimistic projections, if everything goes well, and I think I've said enough caveats now, that there could be efficacy data from one or more vaccines available this fall, potentially more during the winter time. Whether the FDA will allow for emergency use authorization is not known yet, but it is anticipated. And whether the vaccine manufacturers will have sufficient vials available is also not known. So there's many unknowns. So it's certainly not a promise or should not be an expectation. However, those are the most optimistic timelines that people are describing. And that potentially will be for high-risk individuals, potentially first responders or others. It is likely that a vaccine for general use will not be available until 2021, even in the most optimistic scenarios. But those timelines are ever shifting, and so stay tuned. There are no promises, and as we know in science, not everything works perfectly the first time. And that's certainly true for vaccine development. So there is reason for optimism, but there is no definitive timeline yet when a vaccine will be available for the public.
Thank you. I'll ask Megan Murray, there's quite a bit of interest in this wastewater epi approach. And can you speak a bit more about the extent of deployment and how helpful it could be for catching in advance a local outbreak, for example?
Yeah, so Tim Erickson and BioBot have been developing this for a number of years. They've looked not only in the past for pathogens, but for chemicals like opiates and looking for signals for antibiotic resistance. So this is a fairly well-developed tool. It's widely used in Europe for sampling communities for a whole range of different things. So it has correlated very nicely with evidence for the entry of the COVID-19 into different communities. And in fact, Tim pointed out that it picked up in advance of clinical data around the COVID-19 entry into Boston. It picked up evidence that there was significant amount of viral material in sewage several weeks before the numbers started to rise. So it's really a very interesting tool, mostly because it gets this early signal, but also because rather than sample every single person that one is thinking about, obviously people need to be sampled for their own clinical purposes, but for the purposes of figuring out where infection is spreading, and in particular, in this case, where infection spreads prior to the clinical evidence of illness, it's a really great tool. And I think that this team has done a remarkable job of trying to set up the system for getting data from around the country. So having a real-time map of where infection is spreading even before cases are determined is going to be a real benefit to the community at large.
Great, Megan, let me keep you for a second because there's a number of questions about reopening and whether there might be advantages to the BCG vaccine if it is proven effective over other vaccines, because BCG is ready and could be deployed right now. Could you speak a little bit more about that?
Yeah, so that's a complex question. BCG is ready right now in the sense that every year 100 million doses of BCG are manufactured and distributed. BCG has been given to babies at birth in most countries in the world. US has never used that policy, but Europe backed off in the last couple of decades. So we do know that it's possible to manufacture and distribute BCG. But we also know that BCG is essential for preventing childhood TB, and that it's not, although if you can make 100 million doses, it's certainly doable, but it's not trivial to scale up manufacturing even the BCG. It's probably easier than developing some of the new vaccines, but I think if it's proven to be useful, it will be challenging to scale up at a global level. On the other hand, we know how to make it. We know that it's safe because it's been used in, as I say, 100 million people a year receive it, babies. We are fairly convinced that it's safe in older people because of a handful of trials of revaccination that have taken place in a number of countries. But we don't really understand this non-specific effect. And it's been documented in young children. We don't know if it applies to older people. And this is just something that we have to do a clinical trial to see. And in fact, we have to do multiple clinical trials because we really need to see both whether this works in different populations, whether the older people who are at highest risk for bad outcomes will benefit from it. And there's also a number of different strains of BCG. BCG is a live bacteria that has been growing in different labs since the 1920s. So the strains have diverged somewhat. They seem to have slightly different immunogenic profiles. And we're really going to need to see if strains differ and if the efficacy differs in different populations. So I think there's some real advantages to having a vaccine available. We don't want to divert something that we know works for kids and prevents a lot of deaths from TB to a different use until we can prove that that's really going to be beneficial.
Thank you, Megan. A question for Lindsey Baden. Can you speak a bit about the use of convalescent plasma as a treatment?
Yes, no, as discussed during the presentations, harvesting the immune response and antibodies that can attack the virus is very attractive, particularly when made by the adaptive immune system, meaning all of our immune systems. And that is ongoing, and that trial is launched. In terms of when we'll have data, it's a few months away, depending on the burden of the epidemic and the number of cases we have. What I think is particularly exciting about this is it's bench to bedside, but it's bedside back to bench, because from this, we can learn a lot more about protective immune responses and which of these may be playing a role in controlling the infection. So it's a very dynamic and back and forth type of trial, but hopefully we'll have data in 2-3 months, depending on the disease burden in our communities.
Thank you, Lindsey. A question for the co-leads of this whole effort, Arlene Sharpe and Bruce Walker. What are the roadblocks that researchers are experiencing, and how is MassCPR helping to overcome some of these roadblocks?
Well, first I would say that the progress that's been made in 2 months is truly inspiring and could only be made by the collective efforts and collaborations that MassCPR has initiated. I think one of the challenges is that many of us would like to be able to do work at the valuable resource at the needle. And while MassCPR has been able to support new staff scientists there, that additional support would be able to help our work move even faster. I'll turn it over to Bruce.
Yeah, I would agree with Arlene. It's just been extraordinary how the whole entire community has come together, and to be on these tech talk calls, as Galit was referring to, which we have for each of the working groups, is really an amazing experience, because you experience collaborations being launched in real time as people toss ideas back and forth. So it's created an entirely different mode of operation in terms of science in this phenomenal biological, biomedical ecosystem that we have here. I think one of the challenges is that we put out an RFA and we got 448 applications from that RFA. We were able to fund some 60 odd grants through the generosity of the various donors that have been mentioned. But there are many more grants that we would love to fund that have been vetted through an incredibly rigorous process. And so we're vigorously trying to link those opportunities to donors that might want to see contributions they make towards solving this pandemic funneled to really immediate action and funneled towards real results. We're very enthusiastic about the science that we're doing, the technologies that we can bring to bear, and just the quality of the interactions. I know so many more people now here than I ever knew before. It's really been exhilarating, just completely energizing in the face of a pandemic that is really devastating.
Yeah, and I would add to that all of this work and bringing people together really is laying an infrastructure so that we'll be better prepared for the next pandemic by working together.
Thank you, Arlene and Bruce. We have a very provocative question from Min Chen, one of our close collaborators. We have read about the virus spreading to animals in a few cases. And I might add that I believe it was just documented in the New England Journal of Medicine that cats could spread amongst themselves. Should we be worrying about this? And if it's true, how can the control of this mode of infection be exerted? I don't even know who might be best at answering that. Perhaps David Knipe or Galit from the Pathogenesis group, or Ron Corley to talk about what type of studies on animals can be performed at the needle.
I would, if I can make a suggestion, Dan has tested the virus in multiple different animal species, and it certainly does seem that the virus can replicate and spread in some small animals. Whether or not that's going to be a risk to humans, I think, is the big question. And Ron most likely can talk to this point, but perhaps Dan can also talk a little bit about the work he's been doing across mice to ferrets to monkeys to humans.
Dan, do you want to start? Yeah.
Sure, I can just add a few brief comments and turn it over to Ron. It is true that this virus appears to be able to infect many animal species. It does not infect mice, but it does infect hamsters, for example. And it has been shown by other investigators, I believe a group in Hong Kong, that hamsters can indeed transmit the virus to each other, even without any experimental inoculations. So I think that these data raise the specter of the possibility that there might either be or could develop an animal reservoir of this disease. If that occurs, it will be vastly more difficult to eradicate this virus from the human population. I don't know there's any evidence of a definitive animal reservoir of this disease, but the ease of infection in multiple different species and the potential of animal to animal spread, I think raises that as a concern. And so I would think that groups such as MassCPR and even beyond should start working with our veterinary colleagues and start to explore those questions.
Yeah, I agree with you. I think
Ron?
what people are really worried about is pets and how their pets get infected and whether or not they can transmit back to humans again. At this point in time, I don't know of any evidence that pets can transmit back to humans, even though the humans who had the virus clearly infected the pets. And it may have to do with viral load, and I think that's going to require a considerable amount of work to sort that through.
Thank you. Let me raise a question posed by Leon Qi of the Wall Street Journal. This one is about children's cases. New York has identified over 100 cases of children and young adults afflicted by an inflammatory syndrome that was not originally reported out of China. And I'm wondering what we know about the nature of this syndrome, and I would pose that to Lindsey, Baden, or Raj Gandhi of the clinical management group.
Raj, so I think that this inflammatory condition in children is emerging, and it's emerging in multiple countries, in Europe, the UK, as well as the US, New York, and other jurisdictions. And it speaks to the complexity of the pathogenesis of disease. It doesn't seem to be a major form of disease with this illness, but it's a very distinct and important form in a important population, children, the Kawasaki's like, or multiple inflammatory syndrome. And I think we need to understand it better, and it may teach us about Kawasaki's, and it may teach us about the immune dysregulation that this virus can precipitate. Why is it occurring in some jurisdictions and not others? Maybe surveillance, maybe the nature of exposure, maybe genetic susceptibility, maybe cofactors. Many more questions than answers. I think what's important is that the syndrome has been identified, so different communities can go back and carefully look for it to determine if there are genetic, environmental, or other cofactor considerations. And I think that'll be an important lines of investigation going forward.
Thank you. Raj, anything to add to that?
No, only to say that our colleagues in the MassCPR at Children's Hospital and Boston and Massachusetts General Hospital for Children are actively working on this type of problem. It was recognized, as was said, in Europe, but there are cases here in the US, so I think there's going to be intense focus on that. And one of our colleagues in this domain presented at one of our conferences a few days ago. So this is an active area that the MassCPR is involved with.
Thank you very much.
And perhaps I could say, George, this
yes,
is
Galit, please.
That the clinical folks have already linked very closely with the pathogenesis working group to begin to deploy samples to those individuals that can really begin to understand why this disease is manifesting, and more importantly, to identify biomarkers that can help us track the development of this disease and how we can intervene more aggressively. So that initial observation has led to really rapid engagement of the pathogenesis investigators because of the MassCPR consortium.
When Karen Jacobson from Boston Medical Center just momentarily, moments ago, commented on the ability of the consortium to identify less common phenomena like this. That's really the value of that outcomes group and consortium, is as they identify these less common populations, then we can study them effectively. So it's a great collaboration.
Thank you. A question on the infectiousness of the virus. There have been reports that the virus has been detected in semen. Does this mean that the virus can be transmitted sexually? And Raj, I might go back to you with a comment on that.
Yeah, it's a good question. I think obviously the predominant mode of transmission is droplets, and so as to whether there's an independent sexual transmission, we just don't know. There are other instances of viruses being found in semen where the virus itself is not viable. So this too will be determined. It will be, I think, impossible to exactly disentangle it from the droplet mode of transmission, though, since that's obviously through close contact.
I mean, this is Lindsey. I think that with what we've learned over the years is we are surprised. And the seminal vesicles, the testes, are an immunologically privileged environment. So they can get seeded and be hard to clear. And I think with Ebola and Zika, we've been surprised that after the major transmission has been abrogated, that other avenues of transmission were identified. And perhaps several of the Ebola outbreaks over the last 30 years were precipitated by sexual transmission that then started another outbreak. And so I do think this will be a very important line of investigation. But as Raj said, it'll be very hard to disentangle while there is widespread community transmission. And so it'll need to be investigated at the appropriate time. And the reason we do science is we get surprised. These bugs behave in ways we don't expect, and we need to disentangle that to deploy the appropriate countermeasures.
Thank you, Lindsey. I'm going to return to a question from Lian Chi, which speaks about the, and I will answer this and then also ask the group in Guangzhou to give their thoughts. But given the recent tensions between the Chinese and US government, how does that factor into consideration of our collaboration? And I might address the fact that when we were first approached and President Larry Bacco of Harvard University was asked for engagement of our community to support our Chinese colleagues, we knew that there were indeed tensions between the governments. And yet we felt very, very strongly that for humanitarian reasons, that it was essential that we reach out and provide our support. And indeed, I think this is an opportunity to recognize that the virus respects no international boundaries. It is a global pandemic, and it is our obligation to come together as a clinical and scientific community to share ideas. What one needs to confront a pandemic is a pandemic flow of information and an open sharing posture. And I think our relationship speaks to that. There are times when science and medicine are tremendous forces for good, forces for international diplomacy that transcend politics. And I think that's what we have here. If there are comments from Dr. He, and folks in Guangzhou on that point. I invite them now.
Well, I absolutely agree your opinion. For the COVID-19 is the global disease, and for all of them is a doctor and research. We just face the disease. We just I think we work together to against the COVID-19. We don't care maybe the political and maybe tomorrow say A and next day say B. We don't care.
Thank you, Dr. He. Let me ask one. I think we'll have time for one last question, which relates to the nature of the inflammatory response to the virus, which several of the commentators have spoken to. It appears that interleukin suppressive drugs may mitigate the cytokine storm. And so I know there's a number of these drugs under development, and some of those were hinted at by Lindsey Baden in his talk. Patients on immune suppressive drugs are being told that they're particularly vulnerable to COVID-19, but is it possible that they are actually more protected from progression to severe disease? And there are a number of experts in immunology on the call here who could address that. I might start with Arlene, and then we'll invite comments from Lindsey and others.
Well, I think one of the most fascinating aspects that we need to understand about this virus is why in some individuals you see this inflammatory response go awry, the so-called cytokine storm. And so part of it is timing in terms of the development of this. And so we really have a lot more to learn about which individuals are the ones who are at risk for developing this. It may be a combination of different genetics and other approaches. And so I think it's really too early to make definitive statements here. Maybe I'll turn it over to Raj or Lindsey to get their thoughts about patients who are immunosuppressed.
I mean, I think, this is Lindsey, I think it's a terrific question. And it's a paradox to even think of it the way that you've asked the question, but I think it's spot on. And as Arlene said, it's too early to know. I think this is an area of active investigation. I think Karen Jacobs and the outcomes group will look at this to determine if our cancer patients, our chemotherapy patients, our organ transplant patients, our immunosuppressed patients have a different course or not. And I think that'll be a very important line of investigation that will go on. In addition to the IL-6 blockade, there are 4 or 5 other pathways in the inflammasome that are being abrogated that will also give insight into the implications of the comment. So I think active clinical studies and outcomes will look at this. Terrific question, too early to have a proper answer.
I might add that communications with colleagues in Italy, in particular, close colleague Fabio Siccheri, who runs the transplant program at San Raffaele in Milan, alerted us quite early that it seemed that the patients who were immunosuppressed seemed to be getting a milder form of disease. And indeed, patients treated with the B-cell drug ibrutinib for diseases like lymphoma or CLL or graft versus host disease also appeared to be protected from the severest form of the disease. Now, these were just hypotheses from clinical observations, but they merit much broader evaluation in a larger number of patients. And that's part of why the international collaboration that we have brings together ideas, best ideas, wherever they might arise.
And George, there are actually 2 individuals with severe B-cell abnormalities who don't make antibodies who both recovered, again, along, sort of supporting the fact that that maybe antibodies and B cells are really important for preventing disease, but it may be something else that's important for resolution of disease. And I think one of the strengths of the MassCPR is the work that's being done in innate immunity and T-cell immunity, et cetera. And our hope is to figure that out.
Thanks, Bruce. Yes. Well, listen, that's all the time we have. And I want to thank Arlene and Bruce for their brilliant scientific leadership of this effort. I want to thank all of the speakers. I think you've seen what a broad community of experts, clinicians, and scientists that the MassCPR has brought together, all working in concert, together with our colleagues from China and Italy and increasingly others around the world. This is a global problem, and we need global communication, collaboration, and solutions. So I want to thank you all for tuning in to this first MassCPR public briefing. We're looking forward to holding virtual events like this on a monthly basis, and we look forward to updating you on our progress in June. Until then, be safe, be healthy, and be well. Thank you very much.
Okay, thank you.
Thank you.